based on a lecture we are giving for master students, I have kind of tried to put some information together on tree mortality and just try to point on different facts about tree death, what are physiological mechanisms, but also what are still open questions and where maybe more research is needed and where we should go into more details in future. So I want to start first with a kind of introduction that mortality is certainly something very normal and natural during stand development. So, and that's often referred to as self-thinning. That means if you have, at the beginning of a stand development, you have a lot of small trees, and if every single tree grows, then certainly after some time, you will have less trees, which are bigger, but the ones who are lost on the way, they die, certainly. And what I show here is from two different sides, from a temperate forest and from a tropical forest, the so-called self-thinning line or trajectory, where here plant size, which is in principle basal stem diameter here, and here the number of individuals is shown. And that can certainly also be seen as a kind of stand development. So the stand ages from here to here. So we have less individuals, but every single individual is certainly a bit or gets bigger. So that's a kind of idea. And we refer to that as Yoda's law. And you see here, that is Yoda and colleagues. And it's really a very funny thing that this guy was, his name is Yoda. So that's really the number of plants is proportional to the diameter square. That's the original formulation of Yoda's law, but you could also replace diameter by any biomass related information. And so in principle, self-thinning is a density dependent mortality due to competition. If single trees grow, they outcompete their neighboring trees. And that law is true mainly, or in principle, for even age stands. So it gets certainly much more complicated when we go for stands where the individuals have different ages. And the three main principles, and I think you all know that, it's just to show that mortality is something very normal. There is an upper limit or constraint on the total leaf area, biomass, or stem diameter. So that's all related to biomass parameters, which is a kind of carrying capacity of a stand. And you see here, even so, these stands are quite different. The relationship seems to be very similar. So that's the third point. The inverse relationship is something that is more or less general. And certainly the constraints of, or the carrying capacity of an ecosystem causes that mortality by self-thinning is dependent on the growth rate of the competing individuals. So that is one thing, and we can transfer that into a baseline mortality rate. So as we saw, the stand ages and the biomass of the single individuals grows. And so there is certainly a mortality rate, which then depends on the average diameter of a tree in this even age stand. And that's plotted here on the axis. And you see that is really a kind of time axis in a growing stand. And you can here then just plot an average mortality rate. So that has been done by Brian Enquist and colleagues, which gives the average mortality per year at a given average diameter. And so you can calculate that. That's a theoretical value. And now you can certainly compare that to measured mortality rates in monitoring studies. And that could now be different stands with different ages or different diameters, because maybe the trees grew faster. But that could be also the trajectory of a stand over the longer term. So that would be the ideal case. And now, however, we look at a drought event. And just let's assume that this axis is also a kind of temporal axis. And then during that drought event, we have higher mortality than expected, and that would be caused excess mortality. So we have higher mortality than we normally assume, and we can attribute that to additional mortality due to an extreme event, drought event in that case. And when we now look further, we could have longer lasting effects. So the drought event could have a legacy, so that even when the event has been stopped, there is still higher mortality. But what we also might see, and that's all theoretical values, so that's really not real values, it's just an example I plotted here. We could have also lower mortality afterwards, because here many competing individuals died, and then you have back to normal conditions, you have a competition release for the remaining individuals and a lower mortality, and that might level into the normal values after some time. So that is just to show you mortality is something that happens, but we could, by comparing the theoretical mortality based on the self-thinning curve, we can compare that with a real mortality, and then we might see things like higher mortalities during drought events. And I want now to go more into detail about the mechanism which caused this excess mortality under extreme conditions during drought. And I think everybody also knows that map. That also always changes a little bit. The original one was from Allen et al. And Henrik Hartmann and colleagues have added more information. And now I think it's also an online map where additional mortality events are updated more or less in real time. And so you see that mortality is not only occurring where you would normally expect, for example, in Arizona or New Mexico, where it's hot and warm anyway, and any increase in drought would kill the tree there. But you see it certainly also in Europe. You see it also in the boreal biomes, and you also see it in the tropical forests, where even in the moist, evergreen tropical forest, you see drought-induced mortality. And we go into detail later on why this is really something we also need to expect there. It's just obviously not really dependent on the background climate if drought-induced mortality occurs. So that is something from Allen et al. 2010. And here it's just one of these representations where you definitely see you have mortality is dependent on precipitation and certainly on temperature, but also on the duration of that extreme conditions. So let's normally assume that's the envelope of the current climate. So we have sometimes drought events which are quite long and have also sometimes higher intensity together with higher temperatures. But during most of the time, in most of our biomes, we are in that range where mortality does not occur. And the future climate will get warmer, and that is one of the drivers. You see here, this line is really in a way that you can have really dry periods, and if it's cool, then the plants won't be so much affected. So certainly, as you know, air temperature is driving, in addition to soil drought, which comes mainly from low precipitation, is driving atmospheric drought. And so that will be the future trajectory. And the projection is that we have certainly more and more mortality events. And if you now look outside, we had really now 2 dry years in sequence, and this spring is very dry too. And when you look really at the trees, and we have a lot of bark beetle attacks nowadays, so the biotic interaction certainly also comes into play. And that is shown here by Nate McDowell's representation, where that basis is based a little bit on the Allen et al. Scheme, but it does differentiate between the 2 main reasons for mortality, which is on the one hand carbon starvation and hydraulic or symplastic failure. And you see that this certainly depends on the duration of water stress, and this depends certainly on the intensity. And the main assumption is always, if you have high intensity water stress, even only for short time periods, hydraulic failure is normally, or should be normally, from that model, the reason for mortality. If the stress is rather low, but is really long, then carbon starvation might be the most important one. But I feel it's more complicated, and I will walk you a little bit through the different interdependencies between carbon starvation and other traits that plants do have. Just a short, not to bore you, but just to bring you all on the same level. So a few definitions. So hydraulic failure, or the hypothesis, predicts that reduced water supply coupled with high evaporative demand, and that's where the air temperature certainly comes into play, causes xylem conduits and the rhizosphere to cavitate. That means air bubbles are seeding in the water transport pathway, and that stops the flow of water. And then if there is no water coming to the leaves or to any other plant parts above ground, then certainly the plant is desiccating and dying. The carbon starvation, on the other hand, and you know that plants react with stomatal closure to prevent this hydraulic failure, but then photosynthesis is no longer possible. And then if that happens over longer time, the plant's demand for carbon can't be fulfilled. And then there is a point where the plant starves from not having enough carbohydrates. And as you also know, there are different plant strategies to cope with drought. And that's here mainly related to the stomatal reaction to drought. And you see, we have here the class of isohydric plant species, which normally close their stomats very early during a drought. And that leads to the water potential not getting too negative. So because the plant just loses no water anymore. And so then the water potential does not go down much more. It kind of decouples itself from these continuum soil and atmosphere, and therefore it closes the stomats very efficiently, but water potential is going down. And the more anisohydric species, they leave their stomats much longer open during the drought, and that certainly might lead to cavitation, because the water potential goes down. And if there is too much tension on the water soil, as you know, we get a kind of pressure that is lower than atmospheric pressure. And the divers among you might know that when you have a pressure relief, then less air or less gas can be dissolved in a water column. And then when a diver gets up and the pressure releases, then air bubbles. If he does it too fast, air bubbles seed in the blood. And it's a little bit the same as here. Then air bubbles seed in the xylem and they cause clogging of the water pathway. And here, this representation now shows how the xylem pressure that was plotted on the last graph is related to the percent loss of conductivity, which means here 50% means that 50% of the conduits, of the xylem conduits, they have been cavitated. And that is 50% is often a value that is used to compare different plants and different conditions, but at 50%, the plant normally doesn't die. It's more this shaded area, let's say between 60 and often it's at 88%, where there is a very high mortality risk, but a little bit depending on species. And you see there is a kind of range of water potential where there is still almost no cavitation, and then the curve certainly changes, and then you have here, when you have water potentials going down to -4 for beach, for example, then the likelihood that the plant die is very high. But what you also see, that are different provenances of beach. That is one from France, from southern France, where it's rather warm normally. That is one from Denmark, where we might expect cooler and moister conditions. And you see that there has been an adaptation between the provenances. So the one who comes from a warmer climate is less sensitive to cavitation at a given water potential than the one who comes from the more moist climate. That is now from a paper from Hans Lambers, but in principle that's originally from Brandon Coates' paper. And that shows that there is certainly a relationship between embolism resistance and the general climatic condition a plant originates from. And here we have plotted mean annual precipitation. And here it's again this kind of 50% cavitation water potential as a measure of the kind of resistance or sensitivity towards embolism. And what you definitely see, we have here in regions where the general precipitation is lower. I think I have to close a window because somebody is washing cars. Sorry for that disturbance. What you definitely see is that trees which come from a drier climate, they are more resistant. So their 50% embolism water potential is much lower than the ones who come really from more moist climates. So that would be more tropical, wet tropical trees. So there is definitely this relationship. The question is now, what is the reason for that? How can this kind of resistance against embolisms be modified in different tree species, or even in different provenances of one species, as we have seen for beech before? And definitely vessel diameter is a key. So that's really that's just from one plant, and they looked really at the different vessel diameters, exposed the plants to drought so that the water potential went more and more negative. And then they looked at the different vessel diameter classes when cavitation, again, that's a fraction of cavitation. So that's 100%. That's again, this 50% threshold. And you definitely see that when the vessels are bigger at a given negative water potential, the relative amount of all vessels in this size class which have embolisms is higher. So this is really one thing of a plant on an evolutionary scale to adapt. So just to produce genetically fixed smaller vessels, or as an acclimation within the phenotypic plasticity of a plant of a given species to when there is returning and continuing drought stress, so that the vessels of the plants might become smaller so that it's adjusted and acclimated to these conditions. I have said the resistance to cavitation differs among species from different origins, with different precipitation, with different climatic conditions. That's one thing, but the story is much more complex. And that's also from Brandon Coates' paper, cited in the textbook from Hans Lambers. They kind of calculated the hydraulic safety margin. And that's this here. And they just said it's not important at what water potential we have 50% or 88% in that case here, cavitation. Important is what is a normal water potential the plant operates at, and what is this cavitation threshold? And that's plotted here. So the 0 line is now really the water potential under normal growing conditions. And these values here, and maybe let's look only at the deciduous broadleaf plants now here. So that is conifers, and that's a broadleaf deciduous. And you see here, independent, that could be Mediterranean woodland, and that's tropical rainforest. Independent where you look, these kind of safety margin between the normal water potential the plant operates at and this kind of cavitation, 88 cavitation value is always comparable. That means, and that's again now what points to the sensitivity of also tropical forests to drought-induced mortality, as we have seen on the map from Henrik and from Eric Allen, that this definitely can occur in all biomes, and there seems to be a reason for that, because the safety margin is certainly, on the one hand, driven by the size and the structure of the xylem, but it comes with a trade-off. There is a trade-off between water transport efficiency and cavitation safety. So if you have small vessels, the resistance to the water transport is much higher, and that causes certainly a lower efficiency. And if you think of tropical trees which grow fast, which have to transport water high, and where often also the relative humidity in the air is high, they need an efficient water transport system. So it seems like that there is a kind of balance between this water transport efficiency and cavitation safety, which is more or less the same all over the world. And this threshold, this kind of trade-off is shown here in this triangle. So you have on the one hand conductance efficiency, which is mainly conferred by large vessels. But large vessels means per square meter or per diameter of the xylem, certainly less cell walls and therefore less mechanical strength, which is again also bad for cavitation resistance. And on the other hand, that is also negatively related to the resistance to embolism. So you can't have, obviously you can't have all, you can't have on the one hand very high resistance and at the same time very high water transport efficiency. So that's always a kind of balance between, and that's also shown here for poplar plants. So that was saplings, and they looked within one population. So that's not even different provenances. That's just the variation within one population. They looked really at the cavitation resistance. And so they selected less resistant and more resistant individuals. And they clearly could show this trade-off. So the more resistant ones, they have certainly lower vessel diameters, but that comes with a strong reduction of hydraulic conductivity. And it comes certainly with higher wall thickness, which is certainly also more investment, carbon investment in the xylem structure. And so we might assume that these plants are really doing better during drought, but these ones are doing better during normal conditions. And this variation within one population certainly also shows that poplar does not put all eggs in one basket, because it might be that there are long periods without drought, and so part of the population is then very successful, and the other part is successful in the case where, or in situations, in locations where there is more drought events and more drought effects on the plants. So that is just a short wrap up of the hydraulic failure story. So we do have, on the one hand, isohydric and anisohydric species, but as you also all know, there is a lot of variation, and part of them are maybe not really the one and not really the other. And within one population of a species, there might be also ecotypes or provenances which are going more to the isohydric and others going more to the unisohydric direction. So it's not that clear cut, but certainly in general, you can say that isohydric species, they regulate the water potential more intensively, and that prevents them from being too much exposed to embolisms and hydraulic failure. And it's just the other way in the unisohydric species. That means the unisohydric species, they need more a cavitation-proof xylem, which, however, comes with less efficient conductance. The isohydric species, they can have really a more efficient system. However, if they close their stomates, as we have seen before, and as we will see now, there is a high risk of carbon starvation. And that is really depicted here. This kind of dilemma that plants always have. If they want to take up CO2, they have to lose water. And here is a kind of plot where stomatal conductance for many different species is plotted against photosynthesis. And you see certainly the more the stomates close, the lower photosynthesis can be, because at a given stomatal conductance, only a given amount of CO2 molecules can diffuse into the plant and used for carboxylation in the Calvin cycle. Okay, and now that's what I found always interesting and sometimes a little bit difficult to understand, is how is now really drought affecting the carbon relationships. And what people often see only and first is that you see here, that's a time after a drought event has started. So that's 0 is the start of the drought event. And then that's a time how long the drought impacts a tree. And often people saw, when they were looking at the non-structural carbon compounds, they were seeing, okay, it increases. There can't be carbon starvation because the plant has more carbon, more carbohydrates than before. And so I find really that paper from Nate, where he really showed this temporal sequence of processes happening during drought related to carbon balance, very informative. And that's why I just wanted to show you that a little bit more in detail, because it helped me a lot for understanding the different processes and also how different interaction between source and sink happens and occurs. So what normally happens first during drought, and that is quite puzzling for plant physiologists, is that growth ceases first. And that causes certainly a reduced sea demand if we assume that photosynthesis still continues. And this demand is partially, reduced demand is partially compensated by reduced respiration, because large part of the respiration is growth respiration, but still there is certainly still, after growth has ceased, there is respiration still happening. However, this growth decline, together with ongoing photosynthesis, causes that NSC concentration increases temporally, because the demand is lower, but the supply by photosynthesis is still there. Okay, and then photosynthesis only in a later stage declines. And then we certainly have over time a reduced carbon supply, which leads to a reduction of the contents and concentration of non-structural carbon. And on the other hand, we still need carbon, on the one hand for respiration, as you see here, but on the other hand, the carbon demand for other processes like osmotic regulation and refilling of xylem vessels, phloem transport, we need even more sugars, because the water potential is going down, and in the phloem and also in normal cells, this has to be compensated by increasing the osmotic potential by adding more sugars into the either phloem vessels or into the symplast of the cells. And when you now look at the trajectory of these lines, you will have at a given time point a threshold value when there is no carbon available anymore. And that is the point of carbon starvation. So it's maybe important not only to look at one time point, but really to go more into the long-term trajectory after the drought has started of carbon concentration, to understand or to kind of get more information if carbon starvation happens or not. And then it's really an intensive discussion, and I think we will never get a solution for that. But this example, again, from Henrik Hartmann's work, is a very interesting one, because when we say, okay, drought causes carbon starvation, and we have a little bit looked at the mechanism in the slides before, then we should be able to compare the sugar and starch trajectories with a treatment which definitely exposes plant to carbon starvation. And this real carbon starvation, This real carbon starvation treatment was exposing plants to very, very, very low CO2 concentrations. I think they would have liked to go lower than 40 ppm, but the filters were not strong enough. But 40 ppm is almost nothing. And so the plant which are exposed for weeks here to that treatment, they are really carbon starved. And so what you see here now is the trajectories of needle, branch, and root sugars and starch concentrations in these different treatments. So that's a control, that's the drought treatment, and that is a low CO2 treatment. And the last points of the treatments, they are always the time of death. And what you see, that there is a depletion of starch and sugars under drought, but it's not that strong as under the real carbon starvation treatment. There is one exception, that's the roots. In the roots, the carbon depletion seems to be very comparable between the low CO2 and the drought treatment. So the question is now, are the roots the kind of Achilles heel for carbon starvation? And is it really that mainly the roots get not supported with sufficient carbon during a drought event when stomats close, and the rest of the plant is not that indicative? And I think that's very important to look a little bit closer into that. And I think that's the only experiment I will show. We have looked a little bit on the carbon allocation under drought, in that case in beech trees. It has been done in our model ecosystem, MODEC, and we applied certain CO2 to the shoots and looked at the incorporation into the assimilates, the transport below ground, also the recovery of certain C and microbial biomass in the rhizosphere, and respired CO2, just to understand a little bit the transfer of carbon under drought to the different organs. And what we found is that, and that's now the photosynthetic uptake, so that would be the integral below these curves, which tell us something about the initial uptake. And you see certainly the CO2 uptake was bigger in the control plants, which were never exposed to drought, compared to these 2.5 months drought stressed plants. But what you also see is when you now look down to the label recovery in the mycorrhizal root tips, that they arrived really over a long time, certainly much less carbon below ground, also in the microbial biomass and in the soil respiration, which certainly is an integral of the metabolic processes in the rooting zone. That tells us a little bit, yes, there seems to be a problem with the roots. The roots are not any longer supported with carbon, but we still don't know, is it because phloem transport is not working? Or is it just because the roots are not growing anymore, because they are the ones who are the organs of the plants who are really directly exposed to soil drought? And that is something where we definitely need more information on. Another point is pathogens. We talked until now only about processes which are related to the physiology of plants, but certainly, and that certainly less sugar is available for the plant metabolism, but certainly carbohydrates and assimilates are important and are the key for producing defense compounds. And that is then the other side of the medal, also of Nate's paper, showing that the ability to produce defense compounds over time is also reduced, and that certainly makes it easier for biotic agents to attack the plant and to contribute to mortality. And there is a very nice paper from Oliva et al., which is a paper from Martinez-Villalta's group. And they showed a little bit, and the example is only different fungi. On the one hand, infecting the xylem. On the other hand, infecting cells, being either necrotroph, that means just killing cells and then taking up carbon from these cells, or the biotroph pathogens, which kind of tap into the carbon balance of living plants. And what you definitely see, and I think that's what this picture should show you, the plant needs carbon to allocate into defense compounds and into reaction zones. That certainly reduces the amount of carbon available for other processes like osmotic potential, respiration, general basic metabolic functions. On the other hand, the fungi, they destroy cells and they get incorporated also sugars from the plant. So that's a two-sided dilemma. The plant needs more carbon for defense, and it has more carbon because the pathogens, they tap into the carbon pools of the plant. And that is now shown in this figure. And I think I want only to look at the straight lines, not at that dotted line. So that's a trajectory we saw before in Nate's paper. So we have first this increase because growth stops before photosynthesis stops. And then we have a decrease in the NSC content. And we are converging here to the point where there's not sufficient NSC left to maintain the cellular functions. And here the red trajectory is now with a pathogen infection during drought or just at the beginning of the drought. And you definitely see the plant needs more carbon on the one hand, and it also loses carbon towards the fungi. So this trajectory is much steeper. And then certainly we have additional repair costs of the plant. And now when you compare here this point and this point, you definitely see that a pathogen attack during drought accelerates mortality tremendously. So that means carbon starvation, just to wrap that up, stomatal closure and reduced photosynthesis under drought can obviously lead to carbon starvation. The roots might be the Achilles heel of the plants due to reduced transport. What the reason is still not really clear. And the carbon depletion reduces defense compounds availability, and the pathogens additionally deplete the carbon pools. And I think that together leads to much earlier carbon starvation and much stronger risk of carbon starvation. So I'm almost close to the end of the talk, and that is now really the question, what is more important, hydraulic failure or carbon starvation? And I think the headline already shows hydraulic failure seems to be more important. And that's from Henry Adams' synthesis paper, where he really, at that time, 2017, brought together all the experiments where trees have been killed during drought, and where, on the one hand, carbon, NSC concentration, and percent loss of conductivity, that means cavitation, have been measured. And here now on the x-axis, we see the NSC deviation from control. That means here are the non-stress control plants. And on this side, we have higher concentrations compared in the dying plants compared to the unstressed plants. And here we have lower ones. And here is just the percent loss of conductivity. So that means in that, here we are going to hydraulic failure, and here we are going to carbon starvation. And we have now these different sections of that graph where we have the living ones, so no depletion of NSC and no loss of conductivity. We have here the ones who have only hydraulic failure because they have still no depletion of NSC, but a strong percent loss of conductivity. We have here the carbon starvation quadrant, and you see there is no plant or no tree dying only from or with carbon starvation. What we definitely see is that the majority of cases, both comes together. And I think the main conclusion is hydraulic failure can happen alone without carbon starvation, but carbon starvation occurs mostly together with hydraulic failure, or always together with hydraulic failure, and that's really the majority of cases. I think we have also seen that you need, for example, for refilling of cavitated xylem vessels, you need also carbon, and that means there seems to be an interplay in the mechanisms, hydraulic failure, carbon starvation. If you have sufficient carbon and your xylem is cavitating, you might be able to refill or to actively add sugars to the xylem, and then water flows back, and then you can kind of repair embolisms. But obviously, when you have not enough carbon to do that, and both happens together, that's the main reason for trees to die from drought. So I'm at the end of my talk, and I want to thank you very much for your attention.